Hi guys, welcome to the FLHCC group presentation, Spring 2020.
Okay, so we just quickly want to go through the outline of our presentation. So we're just going to start with like the classic review of the background of our project, which I'm sure you guys know very well. Then we're going to go into the hypothesis and the goals for the semester, followed by our cell transfection, which we got started late last semester. Then we're going to go into the Western blot, which we've been doing all of last semester and then this semester. Then we'll go kind of into like the project discussion, followed by where we want to go, directions we want to go for the rest of the semester, and then some of our fun highlights from the semester, and then last but not least, questions.
Okay, so now some background on FLHCC. FLHCC is a rare type of liver cancer that accounts for about 1% of all liver cancers. It's really unique because it consists of a deletion of two parts. So you have your PRKACA gene and then the DNA JB1 gene. And there's about a 400 kilobase deletion on this chromosome 19 that results in the fusion of these two, exon 1 from DNA JB1 and then exons 2 through 9 in PRKACA. Another thing that's super unique about this is that the chimera is not seen anywhere else in the body. It's unique to FLHCC2, and it's common in liver cancers because it's not caused by things such as cirrhosis, hepatitis, or alcoholism. FLHCC also occurs in adolescents and young adults. The average age is about 21 years of age. You don't see it in older people normally. And the only treatment currently available is liver resection or taking out that cancerous part of the liver. There is no chemotherapy that would help with this.
So part of what makes FLHCC unique is the histology. This is something that we've talked a lot about, and I'm sure that you guys have heard, but we're going to review it one more time. So the histology that we are seeing that really sets it apart is all of those collagen banding in that right picture. As you can see in the left, you have a very uniform looking liver and cell pieces, where on the right, it is broken up by the collagen. This collagen sets it apart not only from normal livers, but also from livers that are affected with HCC, which is kind of a subsect of liver cancer.
All right, so I just want to quickly talk through some background information that we got from Dr. Graham at the Mayo Clinic we partnered with. So he performed proteomics on these liver samples that we just talked about. And he looked specifically at the enzymatic activity of the proline pathway. He actually found that if you look at the little orange squares, that the OAT, P5CS, and the GDH1 and GDH2 were actually upregulated in the proline pathway. And so that's actually shunting everything towards creating more of that proline, which proline is an amino acid that is a building block of the collagen that we're seeing in the above pictures.
Alright, so just to kind of tie everything together, so there are three things that we know, which is the chimera, the histology, and the dysregulation of proline. However, this does not tell us like how this disease like occur, or like exactly how they work together to like create this disease or this cancer. And so that kind of leads us to our overall goal, is just to kind of understand like the molecular cause and how these three things ties together to create this disease, to help us find like effective drug treatments to treat it. And on the right here is a schematic that was put together by Dr. Yang. And so far, this is kind of like our big picture of what we believe is happening within FLHCC. So all of these pathways are supported by primary literature, but have not been proven to be true yet. So this is what we think, and it's just something that we've been wanting to test. And so right now, which we're going to talk about later, is that we're testing like Cyclin E. So we're just focusing on like little parts within this pathway to see if we're able to like get data from it. If we do, then we can look at the upstream and the downstream to see how they play hand in hand within different pathways. So that kind of leads us to our focus for the semester. We're just going to, like what I said earlier, we're going to be specifically looking at Cyclin E. And yeah.
Okay, so Cyclin E is super important for us. A past research student found an OCA paper showing how adding proline to these cells upregulated Cyclin E. So you can see that in that first little Western blot in the upper left-hand corner. The N stands for normal, or you didn't add any proline to it, and then the P is the addition of proline to those cells, and you see a much thicker band. So the cell cycle then right there is Cyclin E is important for the G1 to S phase transition. In the cell cycle, what happens is in a normal function, the Cyclin E would kind of go up over time and then get degraded, so you have lower once it moves into the next phase. So once you're at that highest peak, it'll move into the next phase of the cell cycle. But what then we found is that we found another paper actually regarding breast cancer, which showed that this full-length Cyclin E was being cut by serine protease neutrophil elastase, an enzyme called NE, and then it was being cut into lower molecular weight. And what happens with lower molecular weight is that when it's being cut, it's not being degraded all the way. So with that Cyclin E, you'd see it peaks at the top and then would go all the way down towards like level 0, you could say, or like 0, the amount of it. Whereas there'd still be a constant amount of lower molecular weight when it's cut. So then moving on to this next little image of a Western blot, there is 2 different thought processes going on. So you have the purple, which is where you still have that full length, that really thick band, but it's being cut into all those little smaller lengths of lower molecular weight. And then there's the orange where all of your full length is gone. There's no full length Cyclin E, and it's only being cut into the lower molecular weight.
Okie dokie. So I'm just going to talk quickly through kind of what Ailee and Katrina just talked about, our hypothesis. So we're going to start off with what Dr. Graham found is that there's a dysregulation of the proline pathway, which is upregulating that full-length Cyclin E expression. This is then getting degraded by neutrophil elastase, creating all that low molecular weight Cyclin E that Ailee was just talking about. This low molecular weight Cyclin E actually has oncogenic functions, which is why we're seeing all those like the proliferation of cells and all that good stuff. And this is causing the tumorigenesis in FLHCC. So this is data from last semester. As you can see, this, for the most part, is supporting our hypothesis. In the HEC cells, you can see, so the HEC are the human embryonic kidney cells. You can see if you look at the little arrows, the full length in untransfected versus the ones that have the DNA JB1 PRK ACA gene inserted into them, that there's higher full length. However, we're seeing like not as much low molecular weight in it. In the liver samples, we're seeing in the normal, along with that arrow, if you look at that again, the full length, sitting around the 50, there's full length in every single one of them, but only in the tumor are you seeing the low molecular weight.
All right, so this semester we kind of split up because since we had 2 different directions, so we've been really looking into the human embryonic kidney cells, as Tim was kind of mentioning. And we thought, you know what, we have 2 smart people, Hannah and Katrina, so they decided to start over winter break to get things going and do some transfection. And then the goal of that is kind of to have them make some cells and maybe have them give them to us. And the reason why the HEK cells are very important is because since this cancer is so rare, it's only 1% of all liver cancers, there aren't very many samples to go around, and we do not want to be wasteful and just try to do all these tests. And so therefore we are working to make the HEK cells and try to see if they can behave like this liver cancer. And so that's why Hannah and Katrina have been doing that. They did a great job. So basically, Tim, Ailee, and we focus on the Western blots of liver samples, and we included the HEC samples too, just to kind of repeat the results that we've been working on. And so when we do that, we make gels, then we do the actual Western part, then we image it. It's kind of like let's say a 4-day process. And then we do some densitometry, which is kind of confirming our results and kind of doing some number crunching, and then seeing where we're at and see if it kind of relates to our hypothesis. And then Hannah and Katrina are going to get into more of what they've been doing, but just like a general thing, they started with the HEC 293 cell transfection and assisted with the Western blots that we were doing when needed. They help us with the analysis, see where they're at, and then see how their results are helping with possibly making this a usable cell model to study FLHCC. So there's either way, even though we've both had slightly different focuses for the semester, there's been a lot of overlap and support for each other on both ends. So yeah.
So yeah, so we're just gonna talk about like the goals for like the two different group for the semester. So like what Stick said earlier, there's two different routes for this semester. One is me and Hannah doing cell, and Ailee, Sarah, and Tim doing Western. So for the cell culture goal, our goal is to create a FLHCC cell model with like properties as seen in FLHCC samples. And if this cell model is successful, we can then use it to test the relationship of the chimera, the proline pathway, and the low molecular weight Cyclin E. And like what Stick said, we care about creating a cell model is because we have limited samples. And also right now we predict like this disease, FLHCC, is occurring in stages. So with that, we don't know the background of the samples, and we don't know like what kind of stage they're at. So if we were to have like a consistent model, it can help with us give a better understanding of our data and how to analyze it better. Because if we just continue testing on with FLHCC samples that we receive, it might be a little bit harder to interpret since we don't know the background of it. And also for the Western blot goal, their main goal for the semester is to get repeatable data of the low molecular weight Cyclin E that we have been able to detect in the past 2 semesters. However, we don't know why, but for like each semester, we will only get like 2 good blots or like 1 good blot, and then afterwards it stops working. So that's something that they're trying to perfect. They're trying to perfect the protocol, so then we're able to get repeatable data of low molecular weight in liver samples and also in HEC 293 samples.
So the first thing we're going to kind of break down is cell culture. As we've talked about, there's kind of a little separation in the group. So we specifically were working with human embryonic kidney cells, and we did a series of experiments to make sure that our cells are reflecting FLHCC.
So just kind of like a little bit thing of something that past research students have done. So me and Hannah did not start this. Like have been other students that have done cell transfection before us, which was in spring 2018 when they transfected the HEC 293 cells, which is human embryonic kidney cells. And the pictures A through D are taken with a bright field, and E through H are taken with the fluorescent microscopy. And on the right here, there's this pictures showing how cells look like when they're dead and how they look like when they're alive. So if you see something dark, that means that they're dead. And if you see something kind of like clear and white, or grayish, which mean that they're alive. So from the spring 2018 cell transfection, we can kind of conclude that the transfection went pretty well, since a lot of the cells did not die. We don't see a lot of black spots within certain wells, and also a lot of the cells have taken up the DNA. That's why it's fluorescing. So the transfection for the spring 2018 was a pretty success.
So what actually happens when we say cell culturing? We just want to talk through a little bit about what that looks like and what that means. So this is our cell culture explanation. So you start with cells that are in the flask in an incubator. They're just growing. Our incubator is set to very specific things: 37 degrees, 80% humidity, 5% CO2, things that are designed to make sure that the cells are growing at the correct rate. Once these cells have grown to 100% confluency, which is seen in this second picture, this means that there is one single layer of cells completely adhered to the bottom of the flask. This is important because this allows for best cell health. They're not detaching or dying. This is kind of the gold standard where you want to be. Once we have 100% confluency, cells are counted using a hemocytometer. So this means the cells from the flask are removed from the flask using trypsin. So it's an enzyme that kind of cuts away those bonds. They're spun down until you have a compact amount of cells, and then a dye is added. It's diluted and then added to this hemocytometer. So we're counting little blue circles, basically. Katrina and I spent a lot of time doing that this semester in order to get a good cell count. So using that cell count, that is when you can seed into a 6-well plate. So that means you've perfected the dilution and done quite a bit of math, which took us a long time to figure out, and cells are seeded. You want a certain amount of cells so that, as I talked about, you get that one single monolayer and they're adhered to the bottom of the well. Once they're added into this flask, they're given 24 hours to grow. And after that, then we start adding things. So this step is really important, adding transfection reagent and DNA. First, you mix them together. This is a part where Katrina and I spent a large portion of our semester really trying to perfect that protocol in those amounts. So we'll talk a little bit about those in a little bit. So then this mixture, this transfection mixture is added to the cells. They're given 24 hours to grow, and then cells are imaged under both the bright field, which is just like normal picture of cells, and then a blue field. And this is where you start to see that lit up GFP. And then after that, cell lysate is collected from each well. So that means that what was in that well is collected and then frozen down so that our Western blot group can really dive in and start to test those cells.
So this semester, me and Hannah decided to, and Doc doing also, we decided to test different experiments of the methodology part. That's because when we first transfected, so like our first week of transfection cells, like we thought it was a huge success. Turns out after like learning a little bit about a little bit about cells, we realized that those cells were basically dead. So that's why we kind of went on this hunt thing and try to perfect the protocols and methodology to see which set of experiments would give us the best results in cell viability when we transfect them. And the first one is that we experimented with was cell confluency. The second is time collection. The third one is transfection region amount. And the fourth is imaging perimeters. And the fifth is DNA concentrations. And we would talk about them specifically in their respective slides. So
the first of those experiments was cell confluency. So as I talked about before, you really want that single layer of cells across the bottom. So all of these pictures are just of HEK 293 cells in the seeding stage. So this means that they had just been diluted and put into their specific wells. These cells have nothing added, no transfection, no DNA, nothing yet. So these have just been seeded 24 hours. And A through E, they all show increasing confluencies that allowed us to test seeding density. This is really important because if they're too dense, they don't pick up the transfection reagent, they start to die, they detach. If they're too sparse, you will start to see less transfection and they won't be able to hold on to the flask either. So picture C was what we started with. And I know that sounds a little questionable, why is it C? But that was our 100% confluency based off a protocol, based off some spring 2018 things. And this number was 6.25 times 10 to the fifth. So that's the number of cells per milliliter. And then we looked at two confluencies below 100 and two above 100. So A was our smallest confluency at 80%. B was 90, C, as I stated, was 100, D was 110, and E was 120. So each of those have corresponding cell numbers that we looked into. And through this experiment, we really want to focus on E. So this by far has our best seeding density. You have lots of cells, they're not overlapping, they haven't detached. So this 120% confluency is going to be our best option moving forward. This is something that Katrina and I recreated in most of the other experiments you'll be seeing. This allows for best cell health and ultimately help lead to our best transfection rate. And with a good rate, that means we have a better model of FLHCC. Our second experiment has to do with time collection. So as you can see there, these first two images, A and B, we're seeing some healthy cells. These are HEK cells that have been transfected using that transfection reagent, the lipofectamine 2000. And A and B are at 24 hours. Everyone's alive, everyone's doing well. But if you look at C and D, you can see that the cells have started to clump. They're not in that single layer. They've pulled apart and detached, which means that they're dying. And dead cells aren't viable. We can't run tests on them, as you know. And so that was at a 48-hour time point. So from here on out, we had to look at new time collection points, and we kept that 24-hour. We really wanted to see what a day would do. And then we moved down to 36 hours. So 36 hours allowed for a second time point, but with a lot less cell death. So at this point, cell lysation was done at 24 and 36.
Okay, so our third experiment was testing transfection reagent amount. So transfection reagent includes lipofectamine 2000, and the 2000 is just a specific kind of lipofectamine reagent because there's tons of reagents. And the next one is optimum. And what we care more about is the lipofectamine. And lipofectamine is just a common transfection reagent used to increase the transfection efficiency of plasma DNA into like in vitro cell cultures by lipofection. So if there's too little, there might not be enough for each cells to have like the DNA within their cell. However, if there's too much, it can be toxic to the cells and kills them. So we're trying to find like the ideal amount or the ideal concentration that we should transfect the HEC-293 with. So what this lipofectamine does is that it has liposome. So from earlier for the methodology, we have two microcentrifuge tube, one with the liposome, DNA, optimum, one with just liposome and optimum. We incubate them for 20 minutes at room temperature. And what that does is that it creates the lipoplex during that incubation. So after the 20 minutes, we then put it in the 6 well plates, transfected with the cells. And what should occur overnight is that the lipoplex will then go inside, go past through the cell membrane via endocytosis to create the endosome. And there are two end results for this. One is that it releases the DNA, so that the DNA can then be replicated, or else the other is that it can also fragment the DNA, and the DNA will then be degraded by the lysosome. So this is kind of our data from this experiment. So these are HEC-293 cells transfected with the 1 microgram of the DNA and various amount of transfection reagent at 24 hours. So the top row is the bright field images, bottom is the blue field, and each column has different concentration of lipofectamine. The first is A has 6, and then B has 8, and C has 10. So from this, we kind of concluded that 8 microliter was the ideal amount of lipofectamine to transfect with. That is because if we were to compare A, B, and C, B has the highest fluorescent at their blue field. Like C, there's like a lot of dark spots and just like very little green dots. And also for the blue, for A, there seems to be like good cells. However, there doesn't seem to be like a lot of cells expressing the DNA compared to B. So that's why we consider 8 microliter to be the best reagent amount.
The next experiment we did actually happened over the course of multiple experiments. So this really looked at our imaging parameters. As you can tell at the bottom, those images all look very different, even though they're from a lot of the same experience. So the first one, A and B, are done in auto exposure. So this, we just kind of let the microscope program do its thing and clicked yes. As you can see, there's a lot of green haziness, and it's hard to find exactly where the transfected cells are, and just very unclear. So the next step would be image C. And as you can see, there's a lot less of that haze. It's more defined. And this was due to the fact that we changed from an auto exposure to a set exposure time. After much trial and error, it was found that 870 milliseconds was actually our winner. And this just allowed for a much clearer picture. And then if you look at picture D, the tint was set to a certain parameter, which is 1400. This tint allowed for the microscope to more readily collect that green, as you can see, due to the fact that we are using a blue field, which is much different than just like a normal microscope. So from here on out, we use more control with our imaging. We're much more intentional with it, which allowed for clearer pictures, which allow for better results and reflection.
Okay, our last experiment was testing DNA concentrations. The reason why we're testing DNA concentration is because if there's too much, it's toxic to the cells and it can kill the cells. If there's too little, it might not be taken up by the liposome, and they're just not going to be enough for like all the cells in the wells. So we had to find like an ideal concentration where it won't kill the cells and that there's enough for the liposome to take it inside the cell. So if we were to look at these images, A through D and A1 through D1, they are collected and imaged at 24 hours. E through H and E1 through H1, they were collected and imaged at 36 hours. For each column, there's different concentration of DNA. A and E has 1 microgram, B and F has 1.5, C and G has 2, and D and H has 2.5. All of these cells were transfected with the ideal 8 microliters of lipofectamine that we did in experiment 3. And from this, we concluded that 1.5 microgram was the ideal and like the best one that gave us great results. And that is, so how did we come to this conclusion is that we've looked at each picture individually. So for the bright field pictures, we were looking to see if the cells are floating or if they're circular. So if they are, those are not healthy. So healthy cells, like what Hannah mentioned, is that they do not float and they look more like a web. And then for the blue field, we looked at how much cells express the DNA with the GFP. So all of those were kind of taken into account, and with all of that, we decided that 1.5 microgram was the best DNA concentration. So just to kind of conclude for our cell culturing, so each experiment was used to test one property of the methodology to produce the highest rate of transfection of the HEC293 cells with the chimera. So for cell confluency, we noticed that a higher cell density was needed than the recommended amount that was on previous protocol from previous students. And that was the 120% confluency. So if we were seeding, that should happen at a 7.5 times 10 to the 5th per well. And then for time collection, we noticed that 48 hours produce high number of non-viable or dead cells. So that's why we're collecting cells at 24 and 36 hours. For the transfection reagent amount, 8 microliter of lipofectamine was optimal at our decided time points of 24 and 36 hours based on cell health and transfection rate success. Same with the DNA concentrations. We found that 1.5 microgram was the best concentration per well, just based on their cell health and transfection rate success. So with this new ideal framework, the 8 microliters, the 1.5 microgram of DNA, the time collection, cell confluency, we utilize it to test repeatability, and also we also use the new imaging parameters to see if we can get better images.
So with all of those conclusions, as you guys know, they were different tests. They were individualized, and so now we need to put it all together. So these were kind of our results. So with this, A and D are just your beginning amounts. We're using these new parameters. Everything's been seeded. A and D just show after 24 hours. What are we working with? What do our cells look like? And then we added that 8, that 1.5, and after a few hours after transfection, media was changed. We found that this helped reduce toxicity. So just A, you're seeing normal sitting cells. B and E, you can see that we're seeing still quite a bit of circular cells, but especially in B, we're seeing less of that toxicity. They're more just flat and that kind of web framework we're looking for. And then after 24 hours, B and E were collected. And so those were taken off the well plates, cell lysate. We use a little cell scraper, and you can see the corresponding blue images. These images are using the new parameters, and you're starting to see a lot more transfection, which is really exciting. That means some of the stuff we changed is working better, but also you're seeing much clearer, crisper images, which was something we were working on. After 36 hours, C and F were collected. As you can see with those orange arrows, there's still quite a bit of rounding, but overall, especially in F, we're seeing less rounding than we did at 24 hours, which either could mean that these cells have taken up the chimera really well, they're starting to adjust, or the density was better than well C. It is noticeable that there is still rounding though, like we don't want to hide that fact. There's still a lot more tweaking to this protocol that we think could happen. And then lastly, I just want to bring your attention to the blue field image in picture set C. So as you can see, this is by far the highest transfection rate you've seen in any of these photos, which was a big win for us. That's just really hitting home that some of the changes that we're making are working, that they're doing what they're supposed to, that we're actually seeing more cell health, even though we are like trying to infect them with cancer kind of deal. Overall, this was the only replication round that we were able to create because we really wanted to see if our protocol could be replicated or if we were just kind of drawing at straws. Due to COVID, we were not able to replicate it again, but we're really excited for the next set of researchers that will be able to kind of dive more into this protocol and continue to tweak it. And next comes the Western blot.
Yeah, so kind of switching gears here, we're just gonna, I hope you enjoyed Hannah and Katrina with the cell culture there. So for Western blot, I think you guys have seen this schematic before, but we're gonna see it again. So basically we make gels on our own and you put a comb in there and then you eventually pull it out. So in that first picture there where it says gel, it basically means it's like exactly what it means. It's a gel, and then we make little holes in it so then we can load things in there and run them. So we've done combinations of things like liver samples, the hex samples, some of both, and then kind of given different antibodies to compare that. So then before that, all that fun stuff, we load them in there, and then we run it through an SDS gel electrophoresis. So we did some of this in Bio 120, so throwback. But basically what the SDS does, it gives everything a uniform charge. And so that allows us just to look at mass of the proteins that we're looking at. So it really focuses just on that. So we're not looking at a bunch of different factors. And then so it pulls things down. So things that weigh a little bit more are going to stay up on the top because they're going to get stuck. And then things that are a little bit smaller are going to kind of drive through there and get kind of towards the bottom. And then so you can kind of see there with all the bands, the bunch of different colors there with the bands on the right is a ladder. And so we always load a certain amount of a ladder. So it just kind of shows us where things are at. And then this will help us with results later. And then so we actually run, once we're done doing that, we are moving the gel to a blot. So it's kind of confusing. It took me so long to figure this out in the beginning of the project, but basically you make this gel, it's super awesome, but it can't be something that we can put in an imager because it's like jelly kind of, like it's like a form jelly, but it's just not for the imager. So then we put it in, let's see, yeah, the picture on the top right. So it looks like electrophoresis, but it's not that, it's totally different. And sometimes things we can mix the two. And so basically we're saying, hey, gel, get these proteins off of here and now onto a blot. And it looks kind of like just like a really small piece of paper, which will be able to fit in the imager and be able to take everything we do to it later. So that's a huge point that we need to make sure that we nail. And so once everything is on the blot correctly, we verify it. We verify with the gel to make sure it got transferred. And then so once we have all that stuff going on, so we're like a couple days in now. So now on the bottom right here, we have a primary antibody, and then this is where we focus a lot of our attention this semester. So basically, there is an antibody and it's searching. Thank you, Hannah. And basically, so this antibody is saying, okay, where is Cyclin E? It's running around trying to find it. And once it finds it, it binds to it. And it binds to a certain part. And we'll get into all that fun stuff later. And then, so we do that for a day, let it sit, all that fun stuff, wash it. Then now we're on the secondary antibody where we make that fresh every single time. And then so the secondary antibody says, okay, where has the primary antibody attached? I will only attach to that spot. So exactly where Hannah's circling right now. So it's more of a specific thing. And then we let that sit, we wash it, do all this fun stuff. Then we put it in this thing called the imager. It's in room 206, fun stuff. And so we make the settings correct because other students use it besides us. There's specific settings. And then so on the bottom, we hope to get a nice looking bands, and we hope that all these processes worked well to give us a nice picture. Clearly we struggled with that, and that's okay. Yeah, and so then once we get an image, we send it to Dr. Yang, we upload it to the computer, and then we do something called densitometry, where we actually take a box on the computer and go around each of those little bands, and the computer says, okay, there's X amount in these different bands. So it's just another way to confirm what we're looking at, because sometimes it could look like, you know, on the blot here, it could look like the last 5 lanes have a lot more than the first 5 lanes. And sometimes that's not necessarily true. It just may look that way. So that's just kind of how we do that. All right. And then some troubleshooting too. So this kind of is referring to how we have improved our skills in the Western blot technique to kind of help fine tune what we're doing, to maybe fine tune our results a little bit more. So last semester we switched to a 1 millimeter comb. So that's referring to the space in between the 2 little plates when you're making the gels, that like the first part, and it just makes just a different way, just lines things up better. And then loading samples. So then once you use the comb to make all those little holes, and you kind of get in there and use a pipette to put the sample in there, we used 25 microliters because we thought it could fit a little bit better, and if you have extra room, why not? And we got a new power source. Thank you, Concordia Electricity. Sometimes that doesn't work. And so we got a new one. Thank you, Dr. Yang, as well. And also for transfer, we just used a certain type of membrane. So when we transfer it from the gel to the blot, we want to make sure that it transfers well. And we just thought maybe a different one would help. And let's see. And we got new milk, because that helps with blocking other proteins, so the only proteins that you want will get picked up, and when that's expired it might not work as well, so that's always an issue. So now we're over those little things from last semester. Troubleshooting, we really got in depth, and we helped Ailee, and she caught up to speed obviously right away. She's been doing awesome. So this semester we really focused on antibodies, imaging, and transfer, so kind of the second half of that Western blot technique that I was talking about. So with primary antibodies, there's different kinds. I promise you we will talk all about it. You get the whole spiel. So there's different kinds that you can make, and there's ones that pick up certain things differently, and what we're thinking at least. And then for imaging, there's different like substrates you can use which pick up light from the imager machine at 206 a little bit better. So we kind of fine-tune that, and kind of went off last semester how Hannah and Dr. Yang kind of did some troubleshooting tips as well. And then for transfer, we just kind of, again, fine-tune is like the main theme of this troubleshooting. And we really made sure we used the right membrane, we loaded things correctly, we made a nice sandwich so it's all together, they're sticking together, and that's just a nice hint, but we're gonna go over everything step by step, so don't worry.
Okay, so I just want to talk about troubleshooting with the protein transfer. So we don't usually have issues with the transfer, even though I think most of the team does struggle with it and finds it kind of confusing, at least the people I talk to. We had issues with transfer actually on one of the last blots of the semester before we moved online, which was super sad because we were hoping to get one more blot and it just didn't work. So what happened is when you put the transfer into the transfer apparatus, it needs to be facing the right way. So when we did it, we had it flipped the wrong way. So if you look at the picture, you can see the negative and positive charge. So when we put the charge onto, thanks Hannah, the whole cassette, it's gonna transfer the protein from the gel onto the membrane. But if you have the cassette flipped, it's actually gonna take those charges and it's gonna have them going the other way. So instead of the protein moving from the gel onto the membrane, like those red arrows are doing, and thank you, Hannah, once again, it's actually gonna go the opposite way and go onto the filter paper or into the other transfer buffer, and then we have just lost all our samples, which is super sad, and that's why we didn't get one of our last blots, sadly.
Okay, so now moving into primary and secondary antibody, one of the big focuses of this semester. So primary is obviously the thing that goes on first. Primary's job, if you can tell by this picture in the middle right here, it would be the gray Y shape. So what happens is primary, when you add it on, will attach to the targeted protein, and then you can have two different types of primary as well. So you'd have your monoclonal, which would be like our HE12 or HE172. And then you also have polyclonal, which is a little bit different, and I'll get into that in a second. So with the monoclonal antibodies, what happens is that they attach to specific parts of whatever targeted protein you're looking for. For us, that would be Cyclin E. So it's only going to attach to one part of that Cyclin E. So say you still have Cyclin E in your samples, but it's not what that primary normally attaches to, it won't pick it up. Now, with polyclonal, the polyclonal's job is that it will attach to any part of Cyclin E. It's not as specific as monoclonal, and it's a little less picky, if you would want to say it like that. So it will pick up any size of Cyclin E or any type, and it will attach to that, which is why we see different types of images. So for example, if you're looking at our images on the left right here, from week 6 for the HE12, the liver samples, which are supposed to be in the middle of our blot, you're not seeing a very good detection of them. There's not a very good likeness. They're really faint, you can't see it. Whereas in the polyclonal, that bottom week 4 blot, you're seeing a lot more background, or what we call, looks a little bit dirty, if you could say it like that, and it's more dark, but you're seeing also darker banding. It's picking up more of that. Some things that we did different was that, before I get into that, sorry, secondary is then the next step. And what you'll do with secondary is it attaches to the constant region of the primary, or that end part of the Y that you want to call. And so in that image, the primary or the secondary would be the blue, excuse me. And so one thing that we changed with this semester with the secondary is that instead of only sitting on the blot for 2.5 hours in the 4-degree fridge, we decided to move it to overnight because we noticed that with imaging, we weren't getting as good of a signal and it was much lighter. And so we just thought that that secondary wasn't sitting on the blot for long enough in order to pick that up. And secondary also always has to be made fresh, which is something to note. And that is different from the primary because primary can be used multiple times. And then for the sake of polyclonal, we noticed that we liked using it more than once because the more you used it, the clearer our image got and the better it was. It less dirty, or there was less background with the blot. Something else to note with primary and primary that we noticed was that with the monoclonal, the HE12 or the HE172, they were picking up different samples than the polyclonal was. So again, referring to that week 6 blot that you see on the left, the bands that you can see the darkest, so the 2 on the left and then those 2 on the far right, you can kind of see a little bit darker too. The HE172 is much better at picking up our HEK or the HEK293, the cell lines. Whereas in the middle, those liver samples, whether they're tumor or normal, it's really faint. It's not very good at picking them up. Even with the HE12, we noticed that it didn't pick up any liver at all in imaging. Then with the polyclonal though, that's where we, I think, made one of our kind of coolest realizations from the semester, was that the polyclonal was really good at picking up the liver. And we were able to get a really good image from that. You can move on to the next one.
I was trying. There you go.
No worries, no worries. Okay, so now imaging, that second part of the primary and secondary that we had a lot of troubleshooting to do with this semester. So Tim, Sarah, and I all made a new SOP for tips and tricks on how to solve each of these imaging issues, because for some reason it was just a nightmare. And so kind of starting with different aspects of the imaging, what you'll do is you'll add substrate first after you've added your secondary, and the substrate will then react with the secondary, and then this creates a signal that you will see on your image, and that's what gives you the appearance of bands that you need to see. So with substrate, there are two different types. There's femto and pico. So the different types are used at different for different signals, or not different signals, excuse me, different targeted proteins that you're looking for. So pico is used for things like beta-actin or blots that have a very new primary. And you want to use it for blots that have a very new primary, because that primary is so strong. You don't need a strong substrate in order to see that image. You don't need that strong primary to get a bigger, better signal. So you would only use pico for things that you need to image for about 60 seconds or less. Even that's kind of really high. Femto is used for primaries that are much weaker or older, you could say. So it's a much stronger substrate, and it is used to image things like our hex samples or our liver samples. And so if you're going to use an old primary, you want to use femto so that you get the best image. This is also taken into consideration for the polyclonal. So when using the polyclonal, like I said before, we like the results from the second or fourth use because it was much cleaner. There wasn't as much background. And then we would also, because we're not using it for like the first time, you'd want to use femto for that second or to fourth use of the polyclonal. Substrate amount was also changed because we had started with 0.5 mil of substrate, and we decided to go much less, down to like 0.25 mil or less. Because as long as there's enough substrate to cover the blot, we thought that we were still getting good signal, but with too much, it would make the blot really wet, and that's not something that's really great for imaging either. Moving on to kind of the computer problems, we noticed that we share that space. Obviously, it's not just ours. So when the computer was left on for too long, so there's a little box back by the BioRed imager, when that is left on, when we go to image, our blots are either grainy, all white, blurry, like all matter problems, and you then would just have to turn it off and wait. Like there was no way to solve that problem without having to just wait. So right now, kind of moving on to the right with our images, these are all examples of problems that we had that we put into our SOP. So this first one on the left, it's really blurry. You can tell that there's blots, but it's like dirty too, and it's just, it's not a great image of what you'd want. So what you'd probably want to start with doing, for example, is you'd wash it, image again, do it as many times as you can to clear it. Another example of something that we did was this middle one. That one we figured is more similar to our computer problem, because you can obviously tell that there's bands, but it's dirty and also blurry. You could maybe focus the iris or zoom in, but a lot of times it's just the computer. And then on the far right, this is more of a substrate image or issue, excuse me, and where you'd either want to put more substrate because you're not getting a very dark banding, or change the substrate. Say you use pico for this, you may want to use femto in order to get a darker banding. And then another thing you could do is also increase the exposure time. So femto would be used for something like 120 seconds or more. So like example, 2 or more minutes.
So if that doesn't work, then we're basically out of luck and we just have to do it again. And that's why we sometimes are sad in lab. All right, so we're just going to go over the results from this semester. We're going to quickly go over, again, last semester's results. So remember that this, for the most part, is supporting our hypothesis. I think I forgot to say this before, but in the hex cells, we're seeing the full length to be a lot higher molecular weight than compared to the liver, and that's sitting about a 60-65 kDa. And then we're still seeing that low molecular weight on this blot to be at around that 30 to 35-ish range. And then over in the liver blot, you see the full length to be around the 40, 45-ish area, or even up to the 50. And then still the low molecular weight sitting at the 35-ish. And then just a reminder that in the HEC, we're seeing upregulation of the full length in the transfected versus the untransfected. And then in the liver, we're seeing more low molecular weight Cyclin E in the tumors compared to the normal.
Okay.
Okie dokie.
So these were our two favorite Western blots for this semester. It seems that we always get two awesome ones at the beginning and right at the end. And unfortunately, this was our last one, and then Corona just decided to abrupt our awesome data collection. So that was unfortunate. And my roommate's cat isn't here, so I apologize if you can hear him. I'm trying to keep him down and quiet. So anyway, so basically the one on the left is from our week 1 and 2, which is kind of funny because of course we always get the great data at the beginning. And we decided to put this one on because this is a blot where we used a monoclonal antibody. So you can see in the figure it says HE172. So that means that it picks up Cyclin E at one specific spot. So we were like, okay, cool. We thought we got some good data from that. And then also we picked the one on the right. That was our last one for data collection, and that was a polyclonal, which means that it can pick up Cyclin E in like a few different spots. So maybe like the top and the bottom or the side or different things like that. So we wanted to do that to compare each other. And then if you can kind of see in the labeling each of the lanes that they're kind of laid out the same, like N380 is kind of towards the front and then N422. So it's kind of numerical order. So it's a little bit easier for us to compare them side by side and just see how the monoclonal versus polyclonal look a little bit different. So then moving on to densitometry. So this is after we get the image that Ailee was talking about on the imager, and we put it in the computer, put the boxes around it, and say, okay, how much, well, we quantify how much protein is in each lane to try to confirm or possibly disprove what we think we're seeing on our blot. So then let's start with the pink, the nice tall ones. So that's the full length to beta-actin. And again, we use beta-actin to say, let's say, I think we used the example earlier in the semester, last semester was like, so we put like an amount in each one. So then we say, okay, there's 5 Dr. Yangs in this lane, 5 Dr. Yangs in this lane, all that kind of stuff. So it's just confirming that we didn't overload one tumor lane or one normal lane by accident. So it allows us to actually compare what we're doing and allows us to compare it in a, in a more like uniform way and just kind of to confirm our results in a different way. So in that pink, like I was saying, those ones are really tall in the N lanes, which N stands for normal. And so it looks like full length is higher in normal. So that's okay because, you and I, normal people without cancer, like that's okay. And we need that Cyclin E to push us through the cell cycle. And so that's all right. So then what we're hoping is that low molecular weight is higher in the tumor. So that is oh my goodness, this little guy, I'm sorry. So we're hoping that the little purple bands are higher in the tumor, but it's not really looking like that. Like N542 is looking a little bit higher, N423, we did N423 twice on this one because we had 2 different samples, and it looks like those ones are a little bit higher than like T219 per se. And then the low molecular weight to full length average. So that's just like saying in the protein, how much low molecular weight per full length, and that is adjusted to beta actin still, even though it like doesn't say, and it's still kind of looking like we're like not entirely sure. So that's the yellow band I'm referring to. It's kind of hard to tell, but we're kind of leaning towards that normal has a little bit more low molecular weight. So again, not our favorite result, but like we said, coming up, we have statistics. So we'll be able to confirm that with you in a minute.
All right, so this is the data from our last blot of the semester before we all had to go into quarantine. So based off this graph, the data shown partially goes against last semester's data and the hypothesis. For example, the ratio of full length over beta actin goes along with the hypothesis, showing that there is more full length Cyclin E overall in the normal when it's compared to like the tumor samples overall. The ratio of the low molecular weight over the beta actin goes against our hypothesis. However, in the hypothesis, we thought that there should be more low molecular weight in the tumor samples when compared to the normal samples, as shown by like the purple bars, of course. This isn't the case. Normal samples are showing a higher ratio of the low molecular weight over beta actin when compared to the tumor samples. And then for the low molecular weight Cyclin E over the full length, the data is a little more inconclusive whether it supports the hypothesis or not. Meaning like there's a lot more low molecular weight in the samples of like certain ones, and then other ones it's a lot lower. So we're kind of like in between and not 100% sure what that means. However, in the other tumor samples, there are like, there's just less low molecular weight is what we're seeing, at least in this plot.
All right, so moving into some statistics. So we decided to do this last semester. Hannah had a good friend who is doing some statistics stuff at Medtronic, I believe. Correct me if I'm wrong. So we kind of chatted with him last semester and kind of got some ideas with what we're doing from an expert. And he kind of said, just run a one-sided t-test, and we found the links to that. So Hannah and I worked through that last semester, and then I was able to help Ailee with that this semester. And what we want to do with these stats is, okay, like we take the densitometry data and say, is this difference significant? So for example, like is the difference between full length for the normal and tumor, is that significant? Is it matter? Will it be medically applicable, or will it be applicable to patients? So that's kind of what we're doing, and we're trying to just confirm or disprove our results here. And it's just kind of organizing it into another way. And we did stats on these last two the last two blots that we're using for our poster. And so we got those on our poster, so you should see that soon too. We just finished our draft. And then for this, we only did statistics on the liver samples because we didn't have enough beta actin to run a hex. So some of the blots we ran this semester was like one hex, and then it was like normal liver, and then like tumor liver. So unfortunately we weren't able to have enough, and now we know for next semester, run all 10 lanes. So hopefully this won't be a problem again, but totally fine regardless, since we've had a lot of work on the hex transfection on Hannah and Katrina's end. And the beta actin there, again, it just kind of helps compare apples to apples instead of apples to oranges. It just levels the playing field, confirms our results, and that we didn't over or underload any lanes. And then, so looking on the left here, week 2, on average, it looks like there's more full length in normal, more low molecular weight in normal, and more low molecular weight compared to full length ratio as well. But unfortunately, these weren't statistically significant. So that means like the difference, it's not like too significant. So unfortunately, so like these results like maybe weren't our favorite, but they're not statistically significant. So maybe that's on our side. Who knows? And then moving on to the right, same pattern is showing full length and low molecular weight are more normal. And then same with the ratio there. Thank you, Hannah. But there was, the image didn't show up the shape, unfortunately, for some reason. But the middle one, the low molecular weight to the beta-actin, for some reason, that one was statistically significant. And it seems odd because it doesn't seem like it's that, like the bar between them is kind of small. And unfortunately it didn't transfer. But what that means is that that one was significant in the normal versus the tumor. So it's kind of a weird result because we're hoping that the tumor, so we're hoping the pink bars would have more and they would be significant. But then again, we did lose 4 weeks of possible data collection. So this is kind of next semester's thing to focus on getting more reputable data. And something also important to kind of discuss here as well is that every tumor is at a different stage. So when you clump them together, sometimes the data seems a little inconclusive or the data is insignificant. So for example, if you took, there's a sample on the last slide, it's like, let's just say N380. So we took that sample, that's a patient, you know, if you tested that at multiple different times and you just compared N380 to itself, you might find some significant differences in the low molecular weight and full length. So it's just important to recognize that although we're not seeing super significant differences in this, it might for certain patients. And that's just important to notice for the future as we're trying to figure out a molecular cause and maybe a possible drug treatment.
Okay, so some conclusions now with the Western blot. We concluded that there is a difference between the molecular weight in the liver and HEC samples. So in the liver, we noticed that the full length was around 45 kDa and the low molecular weight was from 40 to 35, kind of depending. Whereas the HEK, we noticed kind of all shifted upwards kDa wise. So we'd see that full length higher with from 65 to 70 kDa, and then the low molecular weight, however, be lower at 30 kDa. Also, the expression of full length and lower molecular weight in transfected cells is not consistent with the liver tumor. So like I was saying there, that HEK is showing up higher in the blot with a higher kDa, but it's still the same pattern as the liver samples, depending on whether or not it's transfected. So if it's not transfected, we're getting a couple different bands, which means it's more of a control. Whereas if it is transfected, we'd be seeing that similar banding to liver samples. And we were able to conclude this based on densitometry. Also, in liver tumor, there is less full length and less low molecular weight, which kind of goes against our hypothesis, but based on the graphs that we were just looking at, that was something that we were able to look at this semester. There is inconsistency of data. Katrina mentioned this a long time ago, but at the beginning we were talking about how we really want that repetitive data in order to be able to compare them to each other, because the more data, the larger your data set is, the better you are able to compare things and actually make a conclusive result. And then there is that inclusivity, I'm not gonna pronounce that word, inconsistency of data, maybe due to those different stages as well though. In FLHCC, this isn't proven to be true, but it is a hypothesis that we have. And then like I was talking about earlier, those different antibodies, we were able to conclude attach and react to different samples better. So there's differential detection sensitivity of cell samples versus tumor samples when different antibodies were used. Quick recap, HE12 picks up the HEC sample or HEC cells, but not the liver. HE172 picks up HEC and faintly the liver, not a very good signal. And then the polyclonal picks up the liver, the best of the antibodies.
So future directions, we're nearing the end of our presentation. Sorry, it's just a teeny bit long. Kind of had 2 projects in one. So for cell transfection, we have a couple different ways that we really think we can up our game. And so with cell transfection, we want to transfect at different hours to really recreate different stages of disease. This allows for us to create a more accurate model that we can compare as we're really looking into more data with our liver samples in that regard. We want to transfect with hydromycin, which is an ampicillin resistance gene, we really want to use this because then we can positively select those that have that chimera integrated into their genome. As you saw, not everything is lit up when we transfect, but the majority of them. So we really want to pick those cells out and start to regrow them. And then after we have a new cell line, we want to test that and really compare the results of that to different Cyclin E and Western blots that we already have off liver samplers.
And then for the Western blot future directions, we want to perform additional Western blotting on the FLHCC human liver and HEK samples so we can get that repeatable data of our lower molecular weight pattern that we're seeing. We also want to blot samples made from the cell transfection cultures that Katrina and Hannah were working on. If we're able to do that, that is a huge plus for our group because Sarah had said earlier as well, there are a limited amount of FLHCC samples and we're trying not to blow through those as quickly as possible. So with that cell model, we'd still be able to test, but not run through samples. And then also we want to analyze these different antibodies more. This was a conclusion we came to later in the semester when we started testing the polyclonal, and we would have liked to have tested before quarantine happened, but as the way things were, we'll have to test it next semester and confirm that these results that we were seeing with the antibodies are repeatable.
So just to kind of keep it light and fun here at the end, we just wanted to show some semester highlights. Although our semester was cut short, we did get a lot done. Let's see here. Yeah, Katrina and Tim and I, we went to the Capitol and presented. It feels like forever ago, but it was so much fun and it was just a way to showcase our project. We went to a team lunch, and then the two little screenshots here of Zoom is our quarantine readjustment. And then lastly, just wanted to show some squad support. As you guys know, probably in the CAM group, we're all obsessed with caffeine, and that's how it keeps us going, especially when our results weren't working and we had to redo stuff and spent like almost a full day in the lab. So then Hannah and Tim were RAs, so they were kind enough to share some of their points with us and buy us coffees. And I think Tim bought me coffee sometimes 4 to 5 times a week. And there's a picture of Tim bringing me a Snickers one day. That was awesome. And then in the middle there, the best picture, obviously, is Katrina had an awesome performance towards the beginning of the semester. So Tim and I decided to go and give her some love. And then lastly, again, we kind of talked about it last semester, but Dr. Yang has just been so awesome. She's been responsive at all hours of the day and night, thankfully. And during this quarantine, we've been able to, I think, really connect as a group. And she's helped us with resumes and letters of recommendation and just kind of advice, you know, faith-based and, you know, science-based. And she just really supported us on a bunch of different grounds there. So we appreciate it. And then, of course, pick a question. I tried to include one extra so you guys could kind of pick. Post it on Slack. We're here for you. Happy to answer any questions. Hope you enjoyed.
Thanks for listening.